Good afternoon, students, and welcome to practice problem week set 7 for nanotechnology and agriculture. It's 3 p.m. We should start, but I'll wait for 5 more minutes so that more students can join. I'll keep admitting them as they join this call. So we'll wait for 5 more minutes and then I can start the presentation. Meanwhile, the same set of instructions, if you were attending my previous classes, so what we'll follow the same drill. I will request all the participants to turn off their mic and switch off their video chat. Though this will be an interactive session, it will be an interaction through the chat box since there will be a lot of students joining in this class. So for the smooth conduction of this session, you can talk to me in the chat box. So what we'll be doing again is solving 10 sample questions related to the practice problem week set. So today's uh, lecture set that we are going to follow is from week 7, and uh, you guys must have already received lecture 8 online modules also, and there was also a feedback form circulated for the course, the TA-ship problem solving sessions. So please do not forget to fill that. So it will be a great feedback for me as well as the NPTEL team. And yeah, I'll wait for 5 more minutes to admit more students. Meanwhile, please turn off your video chat and mic, and we'll try to solve all these questions together. And I'll ask you guys to find out the right answer, wait for some time, collect some correct answers, and then proceed. So if you are a previous student of these sessions, then you might be very familiar. But to the new students, so we'll be solving 10 sample questions that you face kind of in the assignment, and I would ask all the students to try to guess the answer or think about the correct answer, and then we'll spend some time learning about the rationale behind or the concept behind the correct answer related to this course, nanotech in agriculture. And then we'll move on to the next question. So we have time of almost roughly around 2 hours. So if we manage to finish within 2 hours, I can take any burning questions or doubt related to this practice problem session or to the lecture session or any other doubts that you can have. Okay. Yeah, I would again like to restate the instruction. So at all times, please turn off your mic and the video chat for smooth conduction of your session. Thank you. Yeah, it's already 3:05. So let us begin the session. I'll share my screen with all of you, and just give me a second. I hope you can see my screen. Okay, wait. Let me just. I hope you can see my screen and the slides changing. And is my audio clear to everyone? Then we can begin the session. You can let me know in the chat box. Type yes or no. Is my audio clear and screen clearly visible? Great, great. Let me switch on my laser pointer. So I'll keep admitting students to this call, and I again want to reinforce the instructions clearly. Please turn off your mic and video chat. All interaction, do not worry, this will be an interactive session, and all kind of interaction with me will be via the chat box, right? And we are here again to solve a practice assignment corresponding to lecture week 7 for nanotechnology in agriculture. The course instructor is Professor Mainak Das, and my name is Ashutosh Behra. I'm a PMRF PhD scholar at IIT Kanpur. So the first question that we are going to solve, the first question is on your screen. I'll read out the question for you guys. In glycogen, dash units are linked by dash glycosidic bonds. So this is the first question for you guys. In the molecule of glycogen, what are the units and what is the kind of bond they are linked with? Kind of glycosidic bonds. Option A, fructose units linked with alpha 1,5 glycosidic bonds. Glucose units linked with alpha 1,4 glycosidic bonds. Alternating glucose and fructose units with alpha 1,4 bonds. Alternating glucose and fructose units with alpha 1,5 bond. What do you think will be the correct answer, students? I'll give you some time to think about it, and I'll encourage all the participants to try to answer. Even if you do not know the answer, it's okay. Just make your guess or based on your understanding, or if you have followed the lecture weeks, you can post your options or the answers in the chat box. I'll wait for a minute to collect some answers, and then we'll see which one of you guys are correct and what is the correct answer, and we'll understand about glycogen and the units and what are glycosidic bonds. Fine. So all the participants, please try to answer. See, this session will be interactive and participative, and it will be beneficial for both the sides. For me at the giving side and you at the receiving end, right? So what units make up glycogen and what kind of glycosidic bonds do you think are present in glycogen? Great, great. People have started answering. Gaurav is saying third option, alternating glucose and fructose. Nirav says B or C. Little confusion. It's okay. It's okay. If you're confused or if you're guessing, even if your answer is wrong, I'm here to clear that confusion. We will understand in depth about the answer so that these concepts get cleared for you while you are in this course and after even after you finish this course. Vishnu Priya Arshita says B. Pradeep is also saying C. Monica is saying B. Madan Kumar C. Oh, there are a lot of answers today. Kumar says B and Abhinami says B. So there are, if we see, there are two groups of answers that students have answered. It's either B or C. People are confused between B or C. Chitranjini says B. Okay. Let's see what is the correct answer. Correct answer is B. Glucose. Glycogen is made up of glucose units and linked by alpha 1,4 glycosidic bonds. So all those who have answered B glucose alpha 1,4 are correct, and those who couldn't, it's okay. But there was a confusion between fructose and glucose. Now let's see what is glycogen. Glycogen is a multi-branched polysaccharide of glucose that serves as a form of energy storage in animals, fungi, and bacteria. So glycogen is the molecule of energy storage, and it is a polysaccharide molecule. Polysaccharide means there are lot, uh, there are multiple units of sugar or saccharose molecules which are joined in a polymer. And it is a branched polysaccharide means the polymer is not linear. At some points of the polymer, there are branchings of molecules. But it is a homopolymer. Homopolymer means it is only constituted of glucose. There is no fructose inside glycogen. So glycogen is made up of only glucose units, but which are branched, which might be linear, and it is serving as a form of energy storage. So in humans, we have liver storing glycogen. Liver is a powerhouse for storing glycogen, and whenever the body needs energy or whenever you go on a fasting diet, liver converts this glycogen into energy. It is the main storage form of glucose in the human body. This is what I was talking about. Glycogen is converted into glucose in the liver hepatocytes. Glycogen is a branched biopolymer with an average chain length of approximately 8 to 12 glucose units. Mani Shankar is saying hello. Hello, hello. Yeah. So glycogen consists of approximately 8 to 12 glucose units, one molecule of glycogen. And it is a branched polysaccharide, right? Glucose units in glycogen are linked through a glycosidic bond. So what is a glycosidic bond? Glycosidic bond we will look at. So you see there is a reaction here which shows joining of two sugar molecules. This is alpha D glucose. This is a glucose sugar, and the lower one that you see is beta D fructose. So fructose sugar. 5-membered ring and a 6-membered ring. Now they join together via the formation of glycosidic bond. Now what is a glycosidic bond? Two monosaccharides. These are called monosaccharides. Two monosaccharides are joined together by an oxide linkage formed by the loss of water molecule. So what will happen? See this position 1 of the alpha D glucose joins with position 2 of beta D fructose. So there is an OH group here, another OH group here. So glycosidic bond, an H2O molecule or a water molecule will be removed out. So an H from here and OH from here will be removed out, and thus will form the bond called glycosidic bond. Such a linkage between two monosaccharide units through an oxygen atom. So the bond will be through an oxygen atom. That is called a glycosidic bond. So if you see OH and OH, so H2O is removed, minus H2O. So this O will form a linkage between two monosaccharide units, creating a glycosidic bond. So these kinds of bond exist between two monosaccharides. So in glycogen, glucose molecules are also connected through glycosidic bond. Is it clear to the students? Now, are you now able to understand what is the glycosidic bond? And glycogen is consisting of glucose multi-branched polysaccharide. Is it clear to the students? Yes, yes. Okay. Now, if it's clear to you guys, I want to ask a burning question now. Can you tell me another important biomolecule where glycosidic bonds are present? This is a question for you guys. In our body or in all living organisms, there is a very important biomolecule which also contains glycosidic bond. What is that molecule? Can you answer? I'll give you a minute to think about the answer. Yes, yes, Aishwarya, you are very correct. So Aishwarya has answered very correctly. It is the DNA molecule. So DNA is a genetic material. Gaurav, correct, very correct. DNA is a genetic material present in all living organisms. DNA or RNA. So in DNA, the phosphate backbone, DNA consists of the phosphate backbone and nitrogenous bases. So the phosphate backbone is consisted of deoxyribonucleic acid. The DNA stands for deoxyribonucleic acid. So deoxyribonucleic acid is a sugar. The phosphate backbones are sugars. And nitrogenous bases are also adenine, guanine, thymine and cytosine. So the nitrogenous bases are joined to the phosphate backbone through a glycosidic bond called N-glycosidic bond. So glycosidic bond is also an important bond present in DNA in all living organisms. So similar kind of glycosidic bond is present in glycogen, the molecule of interest here, consisting of glucose units. Now this is the structure of glycogen that you see in the screen. So if you see, this is a let's say 3 to 5 to 7 molecules unit of a glycogen. So you can see this is a disaccharide polymer and this is a pentasaccharide polymer, and both of them have branched through this linkage. So there are two kinds of linkages present in a glycogen molecule. The simple alpha 1,4 bond. So this is alpha 1,4 bond. If you can follow my pointer, alpha 1,4 bond, the name comes from position 1 of one sugar and position 4 of the another sugar, the next sugar. They are bonded via removal of water via glycosidic bond. This bond is called alpha 1,4 linkage. But if you see this kind of bond, here also there is this CH2OH. This is COOH, HCOOH group. This is ethyl alcohol group. So here OH. So from this CH2OH and OH is removed, and from here another H is removed. So this oxygen forms a bond. Same glycosidic bond, but here the positions are different. This will be an alpha 1,6 glycosidic bond. So in a glycogen molecule, glucose is a structural unit or functional unit, and there are two kinds of bonds present. In linear polymer, there is alpha 1,4 bond, and in branching sites, there is alpha 1,6 bond. So glucose units are linked together linearly by alpha 1,4 glycosidic bonds. Branches are linked to the chains from which they are branching off by alpha 1,6 glycosidic bonds. Between first glucose of the new branch, so this is the new branch, first glucose of the new branch. So if this is the new branch and this is the stem chain, that's what this statement means. First glucose of the new branch and glucose on the stem chain is connected by alpha 1,6 glycosidic bond. While in the same polymer, glucose molecules are linked by alpha 1,4 glycosidic bond. Is it clear to the students? I hope I have made it simple for you guys to understand. What does glycogen look like, and what are these glycosidic bonds? So now you can differentiate well between alpha 1 4, alpha 1 6 bonds, and what how does glycogen looks like. Great, great. Let's move on to the next question. So the second question is, which of the following is a true? It's a true false statement, and it's all about muscle cells. Myocytes are muscle cells. Myocytes are made up of multiple myotubes. Myocytes are also called myofibers, or none of the above. Which option do you think is the correct answer? Let's take a minute and ponder over this question. Think about what are myocytes. So if you see all these options tell us what are myocytes. Which of these definitions do you think is correctly depicting myocyte? I think this is an easy one, and many students would be able to answer this. Are myocyte muscle cells, or are they made up of multiple myotubes, or are they also called myofibers? Yeah, I can see many answers. Gaurav says option A. Roshan is saying C. Neerav, Harshita, Chitranjani, Navina, Umar, Tanuj. Most people are saying A, but some people are also saying C. Avinash is saying C. Dinesh Kumar A. Abhirami is saying A. Amarjeet also answered A. Oh, there are a lot of participants. It's great to see that people are actively participating. Vishnu Priya is also answering A, and Venkat is also answering A. So let us quickly know what is the correct answer. So there are two schools of thoughts. Myocytes are muscle cells, or myocytes are also called myofibers. The correct answer to this question is option A. Myocytes are muscle cells. So majority of all the students answered option A. Very good. And people who couldn't make up the answer, it's fine. It's fine. So let us understand what are all these. So to remove this confusion, what is a muscle cell? What is a myotube? What is a myofiber? Let's understand with this diagram that I'm presenting on the screen right now. Uh, Sorry, these texts have become a little hazy, but just bear with me. So if you dissect a skeletal muscle, or the muscle biceps or triceps, or hamstring muscles that you have in your thighs, so this is a bone, and a tendon is a tissue which help in attachment of the muscle to the bone, and this is the muscle belly, muscle body, or the thick muscular fibrous tissue kind of. And there are, if you cut through these muscle belly body, there are little blood vessels inside these bundles of fibers, and one such bundle is called fascicle, and inside a fascicle there are several other bundles of what we call as myofiber, and these myofibers are coated with sarcolemma, basal lamina, and satellite cell. So myofiber is the smallest. So myofiber is one of those bundles present in a fascicle, and several such fascicles make up a muscle body. Now inside the myofiber is also constituted by myofibril. Fibril are thin bands of muscular elongations. So myofibrils come together to make myofiber. Now several such myofibers make a fascicle, and several such fascicles make up a muscle body. So if you see here, satellite cells are the ones that are present over the myofiber. They convert into myoblasts or cell, which convert into myocytes. Myocytes aggregate and give myotubes, and these myotubes then aggregate and form myofibers. So myocytes are muscle cells which then accumulate and aggregate, elongate to form myotubes, which are multinucleated. You can see many such myocytes are fusing together to form myotubes, and many such myotubes are bundled together to form myofibers. And here are some of the genes that are expressing or help in conversion of these satellite cells into myofibers. And many such myofibers will now make the fascicle, and many such fascicles will make the muscle body. So myocyte is the smallest muscle cell, then forms the myotubes, then we go on to myofibers, and then fascicle, and then muscle body. So this is an overall picture of a skeletal muscle organization. The muscle cell, also known as the myocyte, is the smallest subunit of all muscular tissues and organs throughout the body. So myocyte, also known as muscle cell, is the smallest subunit of muscular tissue. So if you dissect the muscle further and further to most of the nano sizes, the smallest functional subunit of the muscle is known as myocyte. Muscle tissue is a highly specialized type of tissue made up of cells that have as their fundamental properties excitability and contractility. So what are the properties of these myocytes? These myocytes can contract and get excited. So what happens in a when we when our muscle contracts, what happens? There is a signal from our nerve cells or neurons which enters the muscle cell and then gives its command to contract or relax. So the muscle cell or myocyte needs to be excitable because only then the neuron can signal the muscle cell, and it also need to have this contractile property where this myocyte can contract or relax, which leads to muscle contraction or movement. The cellular elements that make up this type of tissue are called muscle fibers or myofibers because of the elongated shape they have. So we just now saw myofibers or myo are made up of myofibrils. So many such cellular elements fuse to make this type of tissue called muscle fibers or myofibers because of the elongated shape they have. Because they look like fibers, they are called muscle fibers. And muscle fibers generally form through fusion of precursor myoblasts into multinucleated fibers called myotubes. So this is what is a myotube. So we saw the process in which multiple myoblasts fuse to form multinucleated fibers called myotubes. So many such muscle cells or myoblasts will fuse together to form a myotube. So that is a myotube, but a myocyte is the smallest subunit of muscular tissue, is the functional and structural subunit of a muscle. And then we move on to higher levels of organization, from myotubes to fibers, myofibers to fascicle to muscle body. I hope it's clear to the students. The answer is myocyte are also known as muscle cells. Myocyte are not the same as myofiber or myotubes. I hope it's clear to the students. You can just let me know in the chat if it's clear to you. Then we can move on to the next question. Yes, great, great. So the next question, Which of the following has application in packaging of orange juice? So now we have a question on food packaging. Which of these has application in packaging of orange juice? Option A, only silver. Option B, only ZNO, zinc oxide. Option C, both A and B. Option D, none of A and B. Come on, let's see what is the correct answer of this. I'll give you guys a minute. Please try to participate. Even if you guess the correct answer or guess any answer, you can just mention in the chat box. Let's collect maximum number of answers and see what is the correct answer. And we'll understand the concept and rationalize behind the correct answer. Let's take a minute and ponder over this question. Which of these have application in packaging of orange juice? Oh, there are many answers. Many answers. Avinash is saying D, none of A and B. And Harshita, Anjali, Tanuj, John, Roshan, Vishnu Priya, everybody are saying C. And Venkateshwar says D. Amarjeet, Pradeep, Gaurav, Neerav, Navina. Most people are saying C. Okay, and some people are saying D. I think somebody raised a hand. Yeah, you can post your question in the chat box, or you can switch on your mic and talk to me. I think it's Venkateshwar. Do you have any question? Venkateshwar has raised the hand, but or did he post in the? Oh, okay, okay. You were just answering. Fine. You can lower your hand. You can lower your raised hand. Yeah. So now, since many people have answered, let's see what is the correct answer. In the packaging of orange juice, both silver and zinc oxide nanoparticles have application. So as many students have answered very correctly, both A and B is the correct answer. Not only silver, we have the application of zinc oxide nanoparticles as well. So let us see what happens in the packaging of orange juice. So let me introduce you to a special kind of nanomaterial called nanocomposites. These were nanocomposites were dealt with in the types and classification of nanomaterials in as early as week 1 or week 2 lectures. Nanocomposites are nanomaterial having multiple properties of different nanomaterials. So nanocomposites are recently used to form the primary packaging material for packaging food items. And why is it so? Nanomaterials improved the barrier properties of packaging by reducing up to 80 to 90% of permeation of oxygen and carbon dioxide. These nanomaterials provide a barrier to the food so that outer air containing oxygen and carbon dioxide is not permeable to the inner food particle. In our case, the orange juice. So the orange juice packed in a material, nanomaterial, inside of a nanomaterial, will not be exposed to the outer air or carbon dioxide. So hence, because oxygen and carbon dioxide are very crucial in leading to growth of microbes and the food going bad, growth of yeast, fermentation. So all those will be prevented. when nanomaterials are implied in the packaging of food. So that is the whole purpose of food industry, to increase the shelf life of food items. We need impermeability to oxygen and carbon dioxide, which nanocomposites can provide up to 80 to 90% reduction in permeation of oxygen and CO2. It can also be used for manufacturing of oils, carbonated drinks, films, and bottles of beer. So in many industries, nanocomposites are being used, such as manufacturing oil, aerated drinks, and beer industry or alcohol industry. Polymer nanocomposites can be made into packaging with enhanced gas barrier properties, reduced oxygen, water vapor, aroma diffusion through the matrix. Yeah, so aroma diffusion is also an important property which comes into consideration while packaging a food material. So, what is aroma in a food? The smell or the pleasant or the present aura of the food, or the smell of the food that captures, is also a part of the taste of the food. So that escapes via many packaging. So if you can design a packaging which can retain that aroma inside the food, then that will be a very plus plus for the food industry, right? So nanomaterials, polymer nanocomposites have also capacity to retain the aroma of the food into the matrix and to maintain food quality and organoleptic properties, hence extending the food shelf life. So in short, polymer nanocomposite have shown to extend the food shelf life of many foods. So if you see what is required in a good food packaging material, it should have antimicrobial function. It should also provide mechanical support properties. So if you have a liquid, you cannot have a non-mechanical. So the better the mechanical property, we have a, if you have seen all these Tropicana real fruit juice packs, they are called Tetra pack because they have so many layers of packaging, and these provide mechanical support to the juices. So we cannot transport liquid otherwise. Optical properties, thermal properties will maintain the food temperature. Environmentally friendly, the packaging material should be environmentally friendly. Aroma barrier to retain, to remain, to keep inside locked the aroma. Gas barrier so that outer oxygen and carbon dioxide may not come in contact with the food, and a vapor barrier. So all these kind of properties make a good food packaging material, which is provided by polymer nanocomposites. Now, this is a research article showing the nanocomposite packaging containing silver and ZNO on shelf life of fresh orange juice. So this is the direct study showing a nanocomposite containing silver and zinc oxide nanoparticles is increasing the shelf life of fresh orange juice. So compared with pure packaging materials, antimicrobial nanocomposite packages containing silver and zinc oxide as an alternative non-thermal technology can extend the shelf life of fresh orange juice up to 28 days. So upon application of silver and zinc oxide nanoparticles, the shelf life of orange juice increased up to 28 days. So you can store the same orange juice more than a month time period now with the help of AG and ZNO packaging. However, a certain concentration of nano ZNO in the packages showed less adverse effects on the sensory characteristics. So sensory characteristic means the kind of taste or sensory perception of the orange juice. So a certain concentration of nano zinc oxide particles in the packages shows less adverse effects on sensory characteristics. It just shows that the uses of nano zinc oxide particles has less effect on how the orange juice should originally taste. So it is not affecting the taste, texture and feel or sensory characteristic of the orange juice. So both silver and zinc oxide are used in increasing shelf life of the fresh orange juice. Is it clear to the students? Did you guys understand? Let me know in the comment box. You can just type yes or no. If you want again, I will. But I'm trying to like simplify things in a way so that you guys will understand easier. Yes, great, 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 great. Let's move on to the next question. Number 4. Which of these is part of a contractile apparatus of skeletal muscle? So another question on skeletal muscle. Which of these is a part of contractile apparatus of a skeletal muscle? Option A, actin filament. Option B, myosin. Option C, troponin tropomyosin complex. Option D, all of the above. I'll wait for a minute. I hope you can see the question on the screen as well as I'm reading it aloud for all of you guys to enhance the maximum participation. Please try to answer. If you have basic knowledge about the contractile apparatus, you'll be able to answer this question. It's not very difficult. Which of these is a part of skeletal muscle contractile apparatus? Is it actin filament, myosin, troponin, tropomyosin, or all of the above? Let's wait for a minute to collect some answers. Wow, so many people have answered. Avinash is D, Anuradha, Rohini. I think all the guys are saying D. Oh, there is a question. Rohit is asking, so what's the pattern of Sunday's question paper? I quite don't know, Rohit. I'm sorry, but I think it will be the same pattern. You will get mostly MCQs. Yeah, the number might not be 10, maybe higher, but you will get MCQs as far as I know. But see, I'm not the one setting the question paper, so and it's totally dependent on NPTEL and the course instructor. So all I can guide you with is these practice problem sessions. So week 8 problem solving session will be next Sunday, my friend Vishnu Priya. So next Sunday will be the last problem solving session for week 8. So there is a reason why these practice sessions are scheduled a week or two behind the actual release of live lectures, because it is a kind of revision. So all these questions are related to that week set. Even if you haven't if you have missed that lecture, or if those concepts are little weak in your mind, these problem sets will re-establish those concepts and you will be stronger. So this is kind of a revision. These practice problem sets are scheduled a week or two behind the actual airing of live lectures for week 8, right? Next Sunday is the exam. Oh, I didn't knew this. Are you sure? So let me confirm. But let's move on with the practice problem session. Let me talk to them. I'll write to the NPTEL team and ask them when should I conduct the problem session. So do not worry. So let's see the answer to this question. And as you guys have all answered correctly, the answer to this question is option D. All of the above are present in the contractile apparatus of skeletal muscle. It seems like you guys have good knowledge about muscle contraction. Very good. Actin filament, myosin, troponin, tropomyosin are all different kinds of proteins present in the skeletal muscle contractile apparatus. So skeletal muscle is composed of cells collectively referred to as muscle fibers. So we saw in some of the previous questions, we saw the organization of skeletal muscle. So eventually these are cells which are fusing together to form myofibrils, myofiber, myotubes, eventually muscle fibers. So these are typically composed of those myocytes. So again here, this is a cross-sectional view of a muscle fiber. But here what we will look at is the banding pattern in the muscle fiber. So if you see this bigger cross section on a single muscle fiber, there are many nucleus because many myocytes have fused together to form this single muscle fiber. And there is something known as a light I band and a dark A band. So we will come to know what are these bands. And if you cut through this cross section, there are multiple mitochondria, and this is covered with sarcolemma. And this a single bundle of myofibril is constituting these different zones which can be visually distinct. So these bands, light band is called I band, and there is a darker A band, and these alternate in pattern. So there are different zones called Z disc, H zone. So all these patterns come along because of the presence of actin and myosin arrangement of the filament on the myofibril. So thin actin filament is accompanied by thick myosin filament. So the myosin head will actually move along the actin filament attached to it, and many different proteins are involved in muscle contraction. That's why we have these different kind of appearances. Each muscle fiber is multinucleated with its nuclei located along the periphery of the fiber. So this is what I was talking to you about. Every muscle fiber is multinucleated because it is formed by fusion of different myocytes, and all these nucleus tend to be on the outer periphery of the fiber. Each muscle fiber further subdivides into myofibrils. So we are looking at one myofibril, which are the basic units of the muscle fiber. These myofibrils are surrounded by the muscle cell membrane, sarcolemma, which form deep invagination called transverse tubules within the myofibril. The muscle membrane sarcolemma, so there is the plasma membrane of the muscle cell or the muscle fiber is called sarcolemma, and each myofibril contains contractile proteins described as thick and thin filaments which are arranged longitudinally into units called sarcomeres. So as I was telling you, each myofibril will have many different contractile proteins, and their appearance will be in the form of thick and thin filaments, and they are arranged into single units called sarcomeres. So here are the different features, microscopically visible features in a single myofibril of a muscle. The A band is a region of thick filament myosin proteins. So this is the A band corresponding to. So these are thick filament myosin protein. H zone is a central region of A band with no overlapping actin proteins when muscle is relaxed. So these blue lines are the myosin protein and the green lines are the actin protein. So H zone is a zone in which there is only myosin. There is no actin. So there is no green line here, and this zone is called H zone. I band is a region of thin filament actin proteins with no myosin. Similarly, complementary to the H zone, there is a zone where only actin proteins are present but no myosin proteins. So you can only see green bands but no blue bands. And such regions are called I band. And what is the M line? M line accessory proteins in center of myosin thick filament perpendicular to the sarcomere. So M lines are certain accessory proteins, and accessory proteins by mean such examples are troponin, tropomyosin, which are present on the center of myosin thick filament. They are not present on actin. They are myosin related thick filament proteins and perpendicularly present on the sarcomere. So that line of proteins is called M line. And what is a Z disc? Z disc is the zigzag line of Z line proteins and actin binding proteins perpendicular to the sarcomere. So this region from one I band to the next I band consists of a Z disc, also known as one functional unit of muscle contraction called sarcomere. So these are the different regions in a muscle myofibril that we encounter. So when you encounter all these terms, I band, A band, Z disc, H zone, so now you will be able to recognize what is it and what band consists of what kind of proteins. So it's very simple. H zone consists of only myosin, I zone consists of only actin, Z zone consists of one I zone to the next I zone. So one I band to the next I band is a Z zone. And in the H zone, there are certain zone called A bands which have only myosin with no overlapping actin. And there is something called M line which is myosin related proteins on the center of myosin filaments. So now it will be easier for you guys to remember what are these different zones, and you just have to picturize the presence of alternating actin and myosin filaments during muscle contraction and relaxation. Now, the contractile apparatus in muscle is composed of actin, myosin, tropomyosin, troponin, and other minor proteins. So, as you guys have all answered correctly, actin, myosin, tropomyosin, troponin are all major proteins present in the contractile apparatus of the skeletal muscle, and there are also many minor. And here this is a enlarged view of a Z disc. Z disc, as I explained, is spanning from one I band to the other I band to the next I band. And I bands are those regions which are devoid of myosin. There is only actin present in I band. So if you can follow these color codings, alpha actinin, these light blue lines are actinin and actin. This green belt is the myosin belt. Red proteins are tropomyosin. There are troponin proteins also. Myomesin, MyPBC, titin, these are all associated proteins. So titin is this big giant protein which holds these myosin filaments. And this is the M line here perpendicular to the myosin filament. And these are myosin associated proteins. And this zone from here to here, which is devoid of any actin molecules, because actin is all these blue color coded regions. This zone will be the H zone. This will be the H zone. This is myosin, and these are actin molecules. And there are other actin associated proteins and myosin associated proteins mentioned in the different color code. So I hope this is clear to you guys. I know it's a lot of information, but once you go back and look at these slides or in the lectures also, or anywhere you just Google it, it will be easier to understand. You just have to remember the alternating patches of actin and myosin, and then you will be able to figure out what are these different regions and bands, right? Is it clear to the students? Just let me know in the chat if you understood it, or do I have to repeat? Great, great, good going. Let's move on to the next question. Dash is the smallest functional unit of striated muscle tissue. It's a related question. Dash is the smallest functional unit of striated muscle tissue. A. Sarcoplasmic reticulum. B. Sarcomere. C. Z-line. Or D. ATP. What do you think is the smallest functional unit of striated muscle? Let's take a minute and just go back to what we just discussed in the previous question. If you can apply a logic a little bit, then you can reach this answer. So I'll give a minute for all of the guys to answer. Let's collect some answers and then we'll proceed. It's not very difficult. It is from the same muscle contractile apparatus. Which of these is the smallest functional unit of a striated muscle tissue? By striated, I mean voluntary muscle. So, so we have different kinds of muscles in our body, right? There are three major kinds of muscles: voluntary muscles, involuntary muscles, and cardiac muscles. So, apart from this question, I just wanted you guys to pay attention to. Voluntary muscles are all striated muscles because we can voluntarily control them depending on our free will. Involuntary muscles are those which are controlled by neuro signals from the brain. We do not have direct willpower or control over them. And there are specialized third kind of muscles called cardiac muscles. Cardiac muscles consist of the heart muscles, and these cardiac muscles start contraction from the time they are formed. So once they are formed, or the heart is formed in a baby, and the heart muscles do not stop beating until death. So cardiac muscles are always contracting and relaxing, while voluntary muscles is based on our will, and involuntary muscles, the brain controls the involuntary actions. So there is a center in the nervous system in humans which control all the involuntary actions. So apart from this question, so can you guys answer this question? Which is the center in the human brain that controls involuntary actions? Let's see who knows a little bit of biology. To this side question, which is the center for involuntary actions in human body? Do you guys know the answer? Okay, I don't it's okay, it's fine. It's fine, I don't see any answer. So let me tell you, if you guys do not. Oh, there is a correct answer. Gaurav, very good. Medulla oblongata. Madhu is also correct. Aishwarya is saying medulla. Correct. Good. Great to know you are also well versed in biology, human biology. Manikanta says medulla. Very good. So medulla is the region which controls involuntary muscle action. And for striated muscle tissue, we have voluntary actions, right? So do you know what is the center in the brain which controls skeletal muscle action? Another side question. Skeletal muscle control center or voluntary actions? Which one do you think is the answer? The region in human brain which controls voluntary movements, voluntary muscles. Yeah, let me just tell you and move on with these with the questions. So the cerebellum in the brain is cerebellar region. So we have cerebrum, cerebellum, and medulla oblongata. Cerebellum is the region which controls voluntary striated muscles. Okay, let's not further delay the questions. And the correct answer is sarcomere for this question. Sarcomere is the smallest functional unit of striated muscle tissue, and all the students have answered very correctly. So, as the definition says, functional unit of skeletal muscle fiber is the sarcomere, a highly organized arrangement of contractile myofilaments. So, majorly actin, which is the thin filament, and myosin, the thick filament, along with other support proteins. And this is again showing uh, how a sarcomere looks like. We just saw it in the previous slide, the previous question. So these are actin lines, these purple, as you can see, purple or violet. Actin protein, thin filaments, and myosin, these pink reddish line, myosin thick filaments. So alternating actin and myosin filaments constitute a sarcomere. And there are certain associated proteins. Titin protein attaches the actin filament to the Z disc. So this is a Z disc, and showing the M line, myosin protein, the actin protein, and this forms, these are the myofibrils which have these kind of patterns, I band, A band again, and these certain many bundles of myofibrils form the muscle cell which forms the skeletal muscle. So these are actually, these proteins give the striated appearance to the skeletal muscle. So by now I think you guys are clear what is a sarcomere. Now what are other options that we encountered? What is sarcoplasmic reticulum? The sarcoplasmic reticulum is a specialized form of endoplasmic reticulum of muscle cells dedicated to calcium ion handling necessary for muscle contraction and relaxation. So specialized endoplasmic reticulum which releases calcium ion based on neural signals is called sarcoplasmic reticulum. Is the it's is the reticulum of the sarcomere. So you can remember it as that. The reticulum, endoplasmic reticulum of sarcomere is sarcoplasmic reticulum. And here again, if we see muscle fiber or a single muscle cell, this is coated on the outside. So we have the nucleus on the periphery of the myofibrils, and on the outside, this is also covered by a net or a mesh work of sarcoplasmic reticulum. And there are mitochondria and myofibrils, and these are the myofibrils. So these sarcoplasmic reticulum form a meshwork or net and connect all the cells as and connect the nucleus to the cell membrane. The function of endoplasmic reticulum is they are ribosomes for protein synthesis, and they connect the nucleus to the cell membrane. Z lines are dark bands across the striated muscle fiber that mark the junction of actin filaments in adjacent sarcomeres. So these are Z lines again. So I'm just ruling out here all the options so that we reach the correct answer, which all of you guys have correctly given. Sarcomere is the functional subunit, functionalist, structural and functional subunit of a striated skeletal muscle. Is it clear to the students? Yes, great. Avinash was asking, are all the students from BSc Agriculture? I quite don't know, but I think there is a mixed population among the audience or people attending these courses. So that is the beauty of these NPTEL courses. So people from any background can attend these courses and earn a degree. So I think most, yeah, people would be from BSc Agriculture, but many people are from other disciplines as well, maybe biology or nanotechnology. So this is a interdisciplinary course. So I think there will be students from um, many fields. Yeah, let's move on to the next question. Question number 6. In tetanus, so it's not tetanus, sorry for the typo. In tetanus, communication at dash is affected. It's about the tetanus disease. Communication at dash is affected. Option A, blood brain barrier. Option B, eye. Option C, neuromuscular junction. Option D, pancreas or liver. What happens in the tetanus disease? This is also going to be a simple answer. As kids, we all must have, in our due course of lifetime, must have at least once taken the tetanus injection, TT injection. We all used to fear the injection, right? So this was one of the first injections that we used to take. I don't know about others, but I have taken it. At that time, I didn't even knew what was tetanus and why a doctor is running behind me with a syringe. But it's a deadly disease, for the hint, and what communication is blocked. Let's take a minute, and I encourage all the participants to try to answer. This is a very easy question. There are already so many answers. Very good, very good. And everybody is unanimously saying C. Neerav, Anuradha, John, Manikant, Avinash, Aishwarya, Tanuj, Roshan, Vishnu, Gaurav, Chitranjali. Everybody is saying C. If you guys are all saying C, then it must be C. So C is the correct answer. In tetanus, communication at the neuromuscular junction is affected. So you guys are all correct. You must have also taken the tetanus injection. Let us learn a little more about what is tetanus. So this is a depiction of a prehistoric era of a disease, and if you just Google search tetanus, you will see images like this. This is a man in agonizing or in pain, and he cannot bend his body. His body is stiff and is locked in a position. So tetanus is an infection caused by the bacteria. So tetanus is caused by a bacteria called Clostridium tetani. So the disease derives its name from the species of bacteria. It belongs to the genus Clostridium. Clostridium tetani is the causative microorganism. When these bacteria enter the body, they produce a toxin that causes painful muscle contractions. So what happens uh, in the tetanus disease? These bacteria will produce a toxin. They will secrete a protein, which is a poison or a toxin, which will cause abnormal firing of our muscles. So all our muscles, the infected individual, his muscle contraction will not be at his own will, and once the muscle has contracted, it will not be able to relax, or once the muscle has relaxed, it will not be able to contract again. That's why the person here shown in the image, his muscles have contracted and it has not relaxed, and even in his will, so voluntary, his skeletal muscles are all like this, and that's why he's locked in this position. Another name for tetanus is lockjaw. This disease is commonly known as lockjaw. It often causes a person's neck and jaw muscles to lock, making it hard to open the mouth or swallow. So here is a severe form of tetanus in which his whole body is locked, or the muscles have contracted and frozen at that moment. Lockjaw is also a common name for tetanus caused by Clostridium tetani. The tetanus neurotoxin is the main notorious player which causes this disease. TENT. What does it do? It binds to the presynaptic membrane of the neuromuscular junction. So synapse is a term. Synapse is a cleft or a gap between two cells. Probably in most cases it is a neuron with another neuron, or a neuron to a receiving cell. So neuromuscular junction is a junction in which, in a synapse, the signal sending cell is a neuron, and the receiving cell is a motor cell or a muscle cell. So motor neuron is the signal sending cell, so signal generating cell, and the receiving cell is the contractile muscle. So what happens in neuromuscular junction? The sending cell the sending cell synapse is called presynaptic membrane. The the membrane where the synapse organ originates. Tetanus neurotoxin will bind to these presynaptic membranes. This toxin will be internalized. By toxin, every toxin is a protein or a modified protein or a carbohydrate or a molecule. So here this protein molecule, tetanus neurotoxin is a protein. It will be internalized or it will go inside the presynaptic membrane and is transported back through the axon until it reaches the central nervous system. So through that presynaptic membrane, that protein will get then transported through the axon to the central nervous system or to the brain spinal cord axis. There it selectively binds to and is transported into inhibitory neurons via endocytosis. So endocytosis is a process in which extracellular materials can be engulfed inside the cell via membrane budding or membrane incision. So these proteins will enter inhibitory neurons via endocytosis. It then leaves the vesicle for neuron cytosol where it cleaves vesicle associated membrane protein VAMP synaptobrevin, which is necessary for membrane fusion of small synaptic vesicles. So what does it do? This neurotoxin will go inside the inhibitory neurons and then there is a vesicle uh, associated membrane protein VAMP. It will go and cleave those. So here is the diagrammatic representation. This is a presynaptic membrane and here is the intact VAMP in normal cases. While in tetanus neurotoxin presence, the VAMP is cleaved. Now this vesicle is containing inhibitory neurotransmitters like glycine and GABA. So these are certain neurotransmitters which when released from the neurons will tell the responding cell to stop whatever its action is. So in neuromuscular junction, if there is release of glycine and GABA, GABA stands for uh, butyric acid. So these are inhibitory neurotransmitters. This will command the receiving cell or the muscle cell to stop contraction or begin relaxation. Now this will only be possible when glycine or GABA is released from the presynaptic membrane. But now the tetanus neurotoxin will cleave the VAMP where this vesicle can no longer fuse with the presynaptic membrane and lead to release of glycine or GABA. So what happens in tetanus due to presence of tetanus neurotoxin? So here it is mentioned clearly, these synaptic vesicles or these vesicles carry neurotransmitter to the membrane for release. So inhibition of this process blocks neurotransmitter release. So this is the main thing that triggers the disease. Presence of tetanus neurotoxin will block the release of glycine and GABA into the synapse where the motor neuron is guiding the muscle cell to contract or relax. Now there is no inhibitory neurotransmitter. That means the muscle will always contract. There is no relaxation for the muscle because the signal, relaxation signal, which is glycine or GABA, is not getting released in the synapse. That's why the muscle downstream will always contract, which leads to lock jaw kind of position in which all the muscles in the body or neck muscles and jaw muscles have contracted and they are not relaxing. That's why the person, the individual that we saw in the picture is in a bent shape and all his muscles have contracted while it is not able to relax. So here again I have mentioned tetanus toxin specifically blocks release of inhibitory neurotransmitters GABA and glycine from inhibitory neurons. So it attacks the inhibitory neurotransmitters, prevents it from release. That's why the receiving cells or the motor cells, muscle cells, will always stay in contracted form and there is no inhibition to that. So relaxation would not be initiated. So that is a summary of how tetanus disease happens and it is a very deadly disease. Mostly these tetanus bacteria or Clostridium tetani is present in rusted surface. That's the reason when while we were kids, we were given tetanus vaccine whenever we fall down or we play with metal objects or iron objects or we get hit, we get scratches from nails. So all these nails, iron objects and other objects, Clostridium tetani is a very commonly found organism on these rust objects. So we must take tetanus toxoid vaccine to prevent tetanus infection, which is a very deadly disease. Okay, is it clear to the students? We learned a lot about tetanus and the neurotoxin, how the disease is progressing, and how does it affect the neuromuscular junction. I hope it's clear to the students. Let me know in the chat. Yes, yes, great, great, good. Yeah, got it. Good. Let's move on to the next question. The next question is about zinc oxide nanoparticles. Which one of the following is true about zinc oxide nanoparticles? Option A, poor antifungal properties. Option B, inability to self-sterilize. Option C, increases mold count. Option D, used for water purification. This is again a true false question. Read all the statements very carefully. What do you think is true about zinc oxide nanoparticles? Is it a poor antifungal? It doesn't have ability to sterilize. It increases the mold count of organisms, microbes, or is it used for water purification? I'll give you guys a minute to collect some answers. My aim is to collect maximum participation from students so that everyone engages, everyone learns, and they're just not idle or inactive. So let's take a minute and allow others to answer. So people who have not come yet come forward to answer. I would encourage all of them. Please come and answer because we are almost reaching to the seventh question. There are 3 or 4 more questions, and it will be the end of the session. And yeah, then answering is always do not feel shy at all. You just have to type the option in the comment box, right? Be confident. Another reason why I have asked all of you guys to turn off your mic and video chat. No need to shy out. You can just type your answer in the comment box. It's okay if it is wrong. No one is going to mind, or no one is going to judge you. We are all here to learn what is the correct answer. And I think this is a question you can readily answer. Read it carefully and just type the answer in the comment box. Let's wait for a minute and then proceed for the correct answer. There are already many answers. I appreciate all of them. Okay, seems like we have gathered good number of answers. Most of them are saying D, used for water purification. Ramakrishna is saying C, increases mole count, while everybody else is saying D. So I think I should reveal what is the correct answer. Correct answer is option D. Zinc oxide nanoparticles is used for water purification. Because all other options, so people who were getting confused about other options, let me clarify it out. All the other options, if you see, is quite contradicting the last option. Zinc oxide nanoparticles has poor antifungal properties. So if it has a poor antifungal property, it cannot be used as a water purifier. Inability to sterilize, it cannot sterilize the water. Increases mold count. So what do we understand when I say mold count? Mold mold is just a fungus overgrowth. So you must have heard the term bread mold, or you must have seen in your households or in bakeries where bread, when kept on when kept outside for, let's say, 4 5 days, stale bread, what we say, there are molds growing on the bread. So those are rhizopus fungus. So molds are nothing but overgrowth of hyphae and colony of fungus. So zinc oxide nanoparticles will never increase the mold count, but zinc oxide nanoparticles can be used for water purification. So it has good antifungal properties, it can sterilize the water, it can decrease the mold count, and it is used for water purification. Not just fungus, zinc oxide nanoparticles, we will learn in the upcoming slides. Zinc oxide nanoparticles have great antimicrobial properties, not just fungus, bacterial infection or contamination on organic matter, everything is handled by zinc oxide nanoparticles. Zinc oxide nanoparticles are nanoparticles of zinc oxide that have diameters less than 100 nanometers. So what is the size of these particles? Lesser than 100 nanometers. And they have a large surface area relative to the size and high catalytic activity. So which is true for generally all nanoparticles. Nanoparticles, due to their small size, have high surface area to volume ratio. It means the amount of volume they provide is much lesser compared to the surface area. The surface area is much higher, much higher, and which also increases their catalytic activity, because the catalyst needs accessible surface for catalyzing any chemical reaction, for binding into the reactants and turning them into the products, or catalyzing any other step. And these are some uses or properties of zinc oxide nanoparticles, which I've mentioned in a pinwheel diagram. So if you follow it carefully, antimicrobial actions of ZNO materials, particular interaction based on unique properties, intracellular responses. So zinc oxide nanomaterials show antimicrobial actions. They can mediate intracellular response inside a microbe and interact based on unique properties present on the surface of zinc oxide nanoparticles with the microbes. Now, metal ion release. So these are some of the methods in which zinc oxide nanoparticles work. Zinc oxide nanoparticles can release zinc plus ions. Now these ions can go and attach to the microbe cell wall or inside the membrane and cause a osmotic disbalance, which will cause the microbe, which will kill the microbe, or plasmolysis. Adsorption is a very good property of zinc particles, where it can absorb many microbes onto the surface. Reactive oxygen species generation. So zinc oxide nanoparticles can generate reactive oxygen species in presence of water. Now these reactive oxygen species can go and harm the kill the fungus or bacterial cells, because ROS or reactive oxygen species are very toxic to living cells. So in water where there is organic matter, where there is fungus growing, wherever there is alga or bacterial infection, zinc oxide nanoparticles treatment will generate reactive oxygen species which will kill all the living organisms, and the water will be sterilized. So the property of zinc oxide used for sterilization of water. Energy metabolism inhibition, it can inhibit energy metabolism of different cells. Lipid peroxidation can oxidize the lipid hydrophilic molecules. DNA replication disruption and DNA break. So these are uh, many diverse functions zinc oxide nanoparticles can mediate when used in water treatment, and majorly showing antimicrobial actions through all these methods. Now this is a schematic showing here zinc oxide nanoparticles in presence of water and oxygen when present treated in water. When treated with ultraviolet light, it converts this H2O and O2 into ROS, or reactive oxygen species, where we'll have OH radicals, H2O2 peroxide radicals, superoxide radicals, and these ROS species will then go and attack the bacterial cell, cause membrane damage and DNA disruption. So you can see H2O2 molecules going inside the DNA, intercalating and causing DNA disruption, OH molecules present on the membrane, causing membrane damage. So this is the method in which ROS generation by ZNO nanoparticles will kill the bacteria or microorganisms if any present in the water and sterilize the water. So often we can see zinc nanoparticles are used in wastewater treatment, where the wastewater here we wastewater use composites of zinc oxide and titanium dioxide nanoparticles. Titanium dioxide will increase the adsorption of organic waste onto the nanoparticles, where zinc oxide can then mediate the ROS method of killing of microorganisms to give a treated wastewater which can then be released into the water bodies like rivers and oceans. So wastewater treatment has a huge potential application of zinc oxide nanoparticles. Zinc oxide nanoparticles are used in nanocomposite membrane filters for wastewater purification. Is it clear to the students? The answer that you all answered correctly. Zinc oxide nanoparticles are used in water purification, wastewater management due to its antimicrobial activity. It's a very good antimicrobial agent. And we just now saw many mechanisms through which zinc oxide nanoparticles mediate microbe killing. Is it clear to the students? I would again request all the participants to please switch off your mic and video chat so that this session will be conducted very smoothly. So any interaction you want to have with me or with the other students, you can type in the comment box. So is the concept clear to the students? We are almost reaching towards the end of the presentation. Yes, great. Nice, thanks. Let's move on to the next question. Question number 8. Which of the following nanoparticles were used for Chinese jujube and strawberry? So this is a memory based question. You can only answer this if you have any knowledge of application of nanoparticles in food storage. So I'll give you a hint. This is for food storage or increasing the life or quality of these foods. So strawberry and Chinese jujube are very high value fruits or fruit foods. Strawberry is very costly. Chinese jujube too. It's not a cheap vegetable or a berry. So nanoparticles are used to increase the quality and shelf life of these items. So which of the following nanoparticles do you think is used in Chinese jujube and strawberry preservation or increasing the quality? Option A, titanium dioxide. Option B, silver oxide. Option C, silver. Option D, zinc oxide nanoparticles. I'll encourage again all the students to try to answer this question. Let's take a minute and collect some answers. I can see there are already many answers. Let's wait for a minute and collect some more answers. Then we'll move forward. Okay, there are some more answers.
and majority, all of them are saying titanium dioxide. Abhirami, Pradeep, Avinash, Tanuj, Madhu, Vishnupriya, Gaurav, John, Amar, Ganeshan, and again, all of them are saying titanium dioxide, and all of you guys are correct. The answer is titanium dioxide. Titanium dioxide nanoparticles are used for Chinese jojube and strawberry preservation and increasing their quality. Due to their excellent physiochemical nature and antimicrobial potential of nanomaterials, they are widely used for pathogenic microbes in healthcare, crop protection, water treatment, food safety, and food preservation. So, not just food safety and preservation, nanoparticles have immense potential due to their antimicrobial potential and modifiable physiochemical nature because they can kill microbes. They are used in healthcare, crop protection, water treatment, and everywhere. So, we already saw some of the examples. So, nanostructures for smart food. This is an upcoming concept in which nanostructure materials or various types of nanomaterials are used for food, food nano safety, food nano packaging, food nano preservation, and food nano functionality. So, providing safety to food or safety to the crops, microbial safety or pest safety from nanostructures or micro sorry nanomaterials. Food nano packaging, packaging of the foods. Nano preservation, preserving, increasing the shelf life of food and functionality. So, creating probiotics and adding nutritional value to the food can be done with the help of nanomaterials. So, in an orange juice, we can add any kind of nanomaterials which will be functionalized with our desired probiotic, and then we can drink the orange juice and get the probiotic as well. So, there is a process called fortification in which general products or general food items that the public consumes is fortified or vitamin enriched or a probiotic enriched food now. So fortification can be done with the help of nanomaterials also. So food nano safety will cover food nano safety against pathogens, allergens, and pesticides, reduce the heavy metal contamination, and inhibit biofilm. So these are some major functionalities that nanostructures can provide. Food nano packaging, intelligent sensing, nano coated packaging, active antimicrobial packaging, eco friendly edible packaging. So there are many packagings nowadays which are very eco friendly. Edible packaging made from uh, made from seaweeds. You must have come across some startups which are now making uh, which are making straws out of seaweed which can be edible, cups made up of edible material from nanomaterials, antimicrobial packaging, food nanopreservation, water treatment, encapsulation, humidity treatment so that it doesn't go bad in humid atmosphere, and functionality, enhancing the physical property of the food, preventing chemical deterioration, and enhancing the nutritive quality of food. So all these aims are met by the usage of nanostructures or different kinds of nanomaterial for smart food. This is a recent concept that has arrived with the help of nanotechnology. Now here the picture that we see is a Chinese jujuberry plant, Chinese jujuba. Titanium oxide nanoparticles used in Chinese jujube strawberry. And what did it do? It reduces browning of the Chinese jujube. So not much aesthetic value. So many of these fruits not just get sold because of the taste. So the food industry also does effort put effort in preserving the aesthetic value of the food. So much brown and blacker berries are less likely to get sold to the market. So titanium oxide nanoparticles reduces browning of the jujuberry. Oh, sorry. Yeah. It slows down ripening of the jujuberry, slows down senescence and decay. So it will slow down ripening, it can increase the shelf life, delay senescence, and the same for strawberry. So titanium oxide or titanium dioxide is the correct answer. Is it clear to the students? Are you able to understand? Let me know in the comments. I think there is a question. Does it not cause anything by adding these? See, studies till now have shown the beneficiary side of adding titanium dioxide uh, for the jujuberries. But as far as I know, see, any application of a chemical or a titanium is a heavy metal, right? So application of such a metal, and titanium is not very easily found in a body. If we use zinc nanoparticles, if we use magnesium, so these are all ions which are used in our body generally. Zinc is a cofactor for many enzymes in our body, but titanium, our body is not naturally rich in titanium. So yeah, I can understand the point. I think there needs to be studies. You might find studies where titanium oxide application could have been showing some demerits also. But mostly in the food industry, for preservation and ripening effects, we use all kinds of substances, right? You must have heard application of ethylene gas. Ethylene gas is a ripening agent. So bananas, generally the bananas that we eat are no longer natural bananas. They are all ethylene gas treated. So very raw and bananas are treated with ethylene gas, and they are made ripe. So we cannot trust anything now. So I think titanium oxide is mostly beneficial, but there needs to be studies. There will be studies which can show its potential risk. So what the scientists and what the researchers need to do is balance the dose of titanium dioxide, or how much dose is beneficiary for the humans for our consumption without causing any adverse side effect. That's what I think will be the right step to it. So I yes, I cannot rule out that it's totally beneficial and not causing any demerit to the human health or body. Yes, thank you, thank you. Let's move on to the next question. Which of these is a plant protein? Another another easy question. Which of these do you think is a plant protein? Option A, albumin. Option C, casein. Option option B, casein. Option C, fibroin. Option D, gluten. Which of these is a plant protein? So all of you guys read the question carefully. Albumin, casein, fibroin, gluten. Which of these do you think is a plant protein? Let me wait for a minute and collect some answers. Do not rush. You can take your time. Plant protein. I must say you must have heard all these names. You must have come across these words. These are all proteins. Albumin, casein, fibroin, and gluten. Which one do you think is from a plant, and which ones are not? Hmm. Let's check some of the answers. Oh, there. Monica, Anuradha, Vishnu, Priya, Pradeep, Venkat are all saying C, fibroin. Okay. Gaurav, Madhu, Amar, Lakshmi, they are saying gluten. Neerav is saying albumin. And Vishnu changed her answer. D. So there are many options saying C, some saying D, some of them also answered A. Globulins. What could be your question? Avinash, do you have a question? Can you post it? I couldn't understand your question. Globulins. What are you asking about globulins? For your information, globulins are proteins. Globulins derive from their structure because they look like globular proteins, and globulins are water soluble. One good example of globulins I would like to give you are immunoglobulins. You must have heard this term. Immunoglobulins are antibodies produced by our immune system against any virus, bacteria, or any pathogen infection. So the antibodies that we use, that we produce to fight off any infection, are from the globin family. They are called immunoglobulins. But it's not related to this question. You just asked the question, so I answered. So, which of these do you think is a plant protein? Now, people have answered. Let me reveal the correct answer. The correct answer is D, gluten. Gluten is a plant protein. If you are pretty active on social media, there are many people in the West are gluten intolerant, lactose intolerant, gluten allergic. So, gluten is actually a plant protein. They cannot eat wheat. Gluten is a protein found in wheat and all kind of pulses and grains and cereals, while rest all the three proteins, albumin, casein, fibroin, are all animal proteins. Albumin, casein, fibroin is not a plant protein. Fibroin is actually the silk fibroin, and silk fibroin is produced by a worm called silkworm. So it's not a plant protein, it's an animal protein. Oh, Abhinash was asking about globulins versus gluten. So I gave you a brief description about globulins. They look globular in structure, and one of the best example is immunoglobulins. The globulins are animal proteins, while gluten is a plant protein. Gluten is found in wheat and all cereal pulses. So let's move on to the explanation. Let's go one by one. Albumin is a protein present in blood plasma. So all humans have albumin in their blood plasma. Albumin helps. What is the function of albumin? To move many small molecules through blood, including calcium, progesterone, medicines, and other hormones. So, transporting them, albumin plays a role. It plays an important role in keeping the fluid in the blood from leaking into the tissues. So, into the extracellular, so in the tissues, wherever there are capillaries, this albumin will make the blood very viscous, and the blood cannot leak into the tissues. That is the function of albumin. And most of us regularly consume albumin in the form of egg whites. So the white egg white is consisted of heavily chicken albumin. It's a protein, and there are many forms of animal albumin: bovine serum albumin, fetal bovine serum albumin. So all these are proteins. The egg whites that we eat of are made up of albumin, and this is the egg yolk. Egg yolk is nothing but fat. but these are mainly constituting albumin proteins. Casein. Casein is a protein found in milk that gives milk its white color. Cow's milk consists of around 80% casein protein. In addition to milk, casein protein is found in yogurt, cheese, and infant formulas, as well as in variety of dietary supplements. So all milk related uh, milk related food items and dietary supplements have the protein casein in them. So cheese, curd, yogurt have all casein protein, and casein is the reason why milk appears white in color. So here is a depiction. So all these cheese, we have milk here, have all casein protein. So it is also an animal protein from the cow. Even human milk contains casein. So albumin done, casein done. Now what is fibroin? Fibroin is an insoluble protein. So fibroin is not a globular protein. Fibroin is an insoluble protein present in silk produced by numerous insects such as larvae of Bombyx mori and other moth genera such as Samia and Bronometa. So fibroin is silk fibroin, as I told, produced by Bombyx mori. Bombyx mori is the silkworm, Indian silkworm. So people must have been confused between this fibroin to be as of a plant origin or an animal origin. So it's true that silkworms produce the silk, but the silkworms feed on the leaves of plants. Certain plants like mulberry leaves, where the Bombyx mori silkworm is cultured. So it feeds on the plant leaves, but the protein that it produces is from its own body. So it looks something like this. This is the silkworm, Bombyx mori, and the cocoon that this silkworm produces outside extracellular secretion so that it can transform inside into a butterfly or a moth. This cocoon is melted and processed into protein called silk fibroin. So this cocoon contains silk fibroin, the silkworm's extracellular secretion. Silk fibroin is a fibrous protein with semi-crystalline structure that provides stiffness and strength. And this silk fibroin is very stiff material similar to spider webs. So spider webs are also made up of proteins similar to fibroin. And gluten finally, which is the correct answer. Gluten is a structural protein naturally found in certain cereal grains. Gluten can trigger adverse inflammatory immunological autoimmune reactions in some people. So this is what I was mentioning, that people in the West, many people have gluten intolerance, gluten allergy, because gluten is a protein found in cereal grains. So it is found in wheat, basically, and can trigger allergic reactions. So all kinds of grains. So we have wheat and we have maize, we have barley. So all these kind of rye have gluten in them. So is it clear to the students? We discussed four kinds of proteins. Three of them, albumin, casein, and fibroin, are animal origin, while gluten is a plant-based protein. So the answer to the question which of these is plant-based. The answer is gluten. Is it clear to the students? Yeah, very well, very well. Good. And the last question of this session, it's about milk clotting. In milk clotting, proteases act on dash to form dash micellar state. Option A, casein stable. Option B, lactose stable. Option C, casein unstable. Option D, lactose unstable. Comment below the correct answer. This is the last question, guys. So people who have not come forward to answer, I will encourage all of them to answer. What do you think will be the correct answer? In milk clotting process, there are proteases acting on dash to form dash micellar state. Let's wait for a minute. Read the question carefully in case if you want to change your options. Let's wait for a minute and collect some answers. Which of these do you think is happening in milk clotting? Okay, let's check some answers. Avinash is saying maybe A. John says B. Ritesh, Vishnupriya, Venkatrao, Priyanka, Monica are all saying B. Amarjit says A. Neerav is saying C. Okay, so most people are saying B, while some are also saying A and C. Let's see what is the correct answer. The correct answer is option C. Neerav is correct. The answer is milk clotting proteases act on casein to form unstable micellar state. Guys, it's pretty easy. It's pretty counterintuitive, right? In just the previous question we discussed, casein is the milk protein. There is no lactose present in milk. I doubt that. Lactose is a sugar molecule. So I want to clear your fundamentals. The difference between a sugar and a protein. Lactose, sucrose, fructose, these are all sugar molecules. While casein, albumin, and all proteases are proteins, right? So in if you think of it like this, milk does not have pre-added sugar. So if we add sugar in the milk, then we have sucrose. But in milk, in original milk, we have casein. Well, there might be some lactose molecules. I do not know, but casein is the main protein involved in milk clotting process at least. And proteases act on this casein protein to create an unstable form of micellar state, which then precipitates the protein out of the milk. Now let me explain what is clotting of milk. Clotting of milk is a result of action of proteases that destabilize casein micelles. So casein protein present in the milk is destabilized with the action of proteases because proteases come and cleave the casein molecules, which are particles present in fresh milk dispersed in a continuous phase comprising of water, salt, lactose and whey proteins. So here, so in fresh milk we have water, salt, lactose and whey proteins, so all kind of proteins. But the milk clotting process will target the casein molecules, casein micelles. The coagulation of milk by renin and other proteolytic enzymes involve specific hydrolysis of a peptide bond in the casein. So the proteases like renin and other proteolytic enzyme will hydrolyze the peptide bond in the casein, thereby causing the clotting of milk. And here is a representation of milk clotting. So this is a micelle showing K casein molecule. Calcium phosphate is also present in the micelle. Whenever there is a clotting enzyme, these are casein micelles. Whenever there is a clotting enzyme like renin in presence of calcium, these caseins are aggregated and form curd. So curd that we eat, the aggregated form of milk, has all these cleaved or inactivated casein proteins. Then residual clotting enzyme will if you keep the curd for a longer time, there will be residual clotting enzyme which will act more on these proteins to break it into short peptides which will taste bitter. That's why we stop the curd or the clotting process in between to obtain curd,
and we do not keep the curd outside or with the in presence of residual enzyme, so that it turns sour to bitter in taste. So process has a huge application in dairy industry. So where does it get used the maximum? Coagulation is the push of climb cliff that turns milk into cheese. So this coagulation process or milk clotting process has a huge implementation in cheese industry. So these are cheese wheels or um, big wheels of cheese that are made in fermentation rooms and milk coagulation rooms. So if you go to Bologna or Italy or in Europe, there are big industries which produce cheese. Even in India, Amul is the greatest manufacturer of cheese. So they have all these kinds of techniques for coagulating the milk, for aggregating the casein protein, to creating curd, to creating milk and other milk products. So the pizzas that we eat are loaded with these kind of cheese is all possible due to the process of milk clotting using protease enzymes, right? Liquid milk is converted into a solid mass. The solid mass is often called curd, gel, or the coagulum. So the curd will be used as a coagulum or for inoculation for making of the processed cheese. Coagulation can occur in a few different ways: enzyme action, or acid heat addition. So enzyme action with the use of proteolytic enzymes like renin to cleave the casein protein that we saw in milk clotting. So all these processes are used in majorly in dairy and cheese industry, cheese and other products industry. So the answer to our question is milk clotting process proteases act on casein protein and turn it into an unstable micellar form. Is it clear to the students? This is the final explanation. If it's clear to the students, please let me know. Great. Thank you. Yeah, I can read some of the comments. So if nobody has any questions, I'll just end my presentation here. So let me just stop presenting. Yeah. Yes, students, I'm trying to read your comments in the chat box. Neerav is asking next I didn't knew of this fact that next Sunday is the exam. I will write to the NPTEL people to see when we can schedule this week 8 problem pattern. So these are again exemplar questions. So these are not. Yeah, I know because see the professors in the NPTEL course, they are all they have all recorded these lectures. So whatever you're getting are recorded lectures. Maybe let me write to them. I'll write to them. If I write to them, I hope they will reply because uh, yeah, I think so. I will ask them if the exam is next Sunday and our session is scheduled, then when can we shift the session? So you will get an email from the NPTEL team when the session is scheduled. So do not worry. I will try to like conduct the week 8 session. And thank you for all these comments. Avinash is writing. It became easy to understand. Yeah, great. See the feedback is like encouraging me a lot because I have put efforts in making these slides so that the answers and all are very simple for you guys to understand. So some people might feel that 10 questions for 2 hour session is too long, but this is what I have been prescribed, that you solve 10 questions and you take it very slow and try to address questions of students. And yes, all the recordings, if you have missed any lecture, if you have missed any practice session, so all these will be available via the YouTube link. You must have been provided with the NPTEL link. If you wait a minute, I'll just copy paste the YouTube link here, which contains the playlist for all the recordings. If you want to revisit any recording, you can. Even all these slides will be provided to all the participants in this course. So you can access the slides via the Google Drive link. So I have been updating these YouTube videos. Let me just open my YouTube and I'll copy the link. The playlist contains all the recorded lectures, uh, all the recorded lectures for just so all the recordings of the sessions. Even this session is getting recorded. All these recordings will be put on YouTube, and you will get the link. Let me just paste it here. Yeah, I think this is the list. This is the link for all the. I think this is the list for all the recorded lectures, like recorded practice sessions. Gaurav, Gaurav is saying, I haven't been able to complete week 1, 2, and week 3 assignments as I had to attend a funeral. I had already sent an email to the support team. It would be very much helpful if you could initiate the same. To the NPTEL support team. See, Gaurav, I don't think I can help you in this matter because I am not a part of the NPTEL support team. So all I can do is ask for the exam thing. I can do because my schedule, the scheduling of week 8 session, practice problem week 8 is coinciding with the exam timing. Then I can shift it, or I can ask the NPTEL people. I am another a student just like you guys, so I'm not a sir as per se. I'm a student as per you guys. I'm a PhD student at IIT Kanpur, so I have been instructed to take questions in this course. So I'm sorry to hear that you couldn't complete your assignments. But yeah, NPTEL team should respond. I do not know. If there is a helpline or number or something, then you can contact. Or yeah, I don't think I will be able to help you in this regard. Need of exam pattern, I think exam pattern will be the same. You will get MCQs probably. That's what I somebody asked in the starting of the session. You'll probably get MCQs, the same pattern you were solving in the assignments, but they might not be 10 in number. Yeah, I think so. I do not know quite. There'll be MCQs, so it won't be like subjective questions where you have to write answers. It would be the same pattern. Avinash, providing questions in PDF. No, this won't be. This will be too much for work for me. You can visit the YouTube sessions, right? Or you can see if you get the link, Google link for all the slides. Then all the slides contain the questions as well as the explanations for answers. I think you should be able to take out questions from that instead of me doing that for you. Yeah, so that's it. We have come to the end of the session. So I will talk to the NPTEL. Yeah, Hema Durga, I think you have a question. You can post it in the chat box. Hello. I think that was a that was a mistake. Yeah. So let's end this session. Can you send the link again? Sure, sure. Here is the link to all the playlists. So the playlist contains 6 lectures, and I'll upload this recording as well. You can find it. And you must have gotten an email from NPTEL during the start of these practice sessions. You just go back to February, kind of March, kind of date, and you search for problem practice session. There will be a email containing the Google spreadsheet for all the YouTube lecture links, YouTube recording links, and a Google Drive folder which contains all the slides that I have been presenting in these lecture sessions. So you will get all the practice session questions and everything. Likhit and Abhinash, I have to confirm with the NPTEL team. For now, I cannot confirm. So let's see if I ask the NPTEL team, I get some response. So I'll ask them about the exam timing, the next week 8 session, and the exam pattern. Okay, I can you rely that on me? Let me ask them because even I am not sure about what will be the exam, but it will be definitely MCQs. It would not be subjective. You do not have to worry about writing long answers or explanatory answers. It will be MCQs. And if you have gone through the course nicely and you have attended the practice sessions, then it will be very easy for you guys. So there will be 4 options. You just have to choose, just like we were solving. Just have to rule out and choose the correct answer. Yes, thank you guys. With that, I'll end the session and hope you enjoyed the class. Yes, I'll end the call now. Take care. Have a good evening.